Epigenetics is something that speaks quite wholeheartedly to myself. A majority of my PhD work has been on epigenetics and specifically in dairy cows. So I just want to share with you in more of a general sense because I understand that people have a lot of different, I guess, background on epigenetics, as in they might know nothing at all to that they know a little bit about it. So this talk is just a very general talk on some interesting studies out there, trying to get your mind thinking about it. So how many people, just a raise of hands, know what epigenetics is or have heard the word before at all? Okay, this is pretty, yeah, so you got like about 50% of the room, maybe about 60% of the room. Epigenetics has been gaining a lot more attention in the popular media, whether it's why your DNA isn't your destiny, or National Geographic's about why identical twins might not be quite so alike, or how the first 9 months of your life actually shape the rest of your life in general. It has implications not only in disease pathways, but also in the formation of memory, aging, and cancer. So epigenetics is one of these hot topics in human health, and we can definitely see some applications that might come into the agriculture industry. So if we're going to get, if we're going to talk about epigenetics, we really need to start with Genomics 101. So the one thing that all life has in common is that it has DNA. And some of the things about DNA is that DNA is made up of 4 base pairs, which can be displayed in a number of different a lot of different sequences, which help tell what proteins are going to be transcribed by that species. So looking at these 2 DNA sequences, do you think you can tell which one's a cow and which one's a human? Not really, no. It might be this, or it might be that. But what's really unique about it is that these sequences offer the history of a species. So it's not only telling the past, but it is also telling the past, the present, and the future. And so they're really like an archive of what those species have gone through. Some interesting things is that we're very similar to mice, actually. We share a lot of our same genome, but mouse is very different from ourselves. And so some of the questions that you should be thinking about, well, mice have 30,000 genes, and we have relatively about the same as well too. Why is a mouse, and why is a human, and why is a cow? So I'm going to go through briefly, some bigger concepts on terms of what epigenetics is. So epigenetics literally means above genetics. Epi is Latin for above. And really this is the instruction manuals of life. So you can think of genetics as the words in a book, and those are the proteins that are going to be expressed by that individual. Epigenetics is how that book is presented, so the order of those words and the emphasis within that book. And overall, if you have a collection of encyclopedias, basically, which I don't know if everyone knows what those are, since those seem to be a dying thing. But if you have the encyclopedia of an organism, that each one of those books represents a certain cell type, that's what really brings out the overall phenotype of those organisms. So it's the instruction manual of life. So with the raise of hands, do you think these 2 individuals are identical twins or not? So identical or not identical? So we got about a 50-50 split. And what about these 2 ladies? Identical or not identical? So these individuals are actually identical twins. So this is kind of where some of the thinking started to come out of what's happening within a species with epigenetics. So some of the unique things about identical twins is that as they age, they become more different. So we know identical twins share the same genome, but as they get older, their disease susceptibility, you can see here how the architecture of their face is. You can have an obese individual or identical twin as compared to to their twin individual. And this is really feeding back to this changes in epigenetics that you might accumulate over time. It might be partially due to genomics as well too, but epigenetics is a little bit more rapid in bringing in that environmental information. So now thinking about cows, we know that these cows are probably genetically different, but they might actually share some of the same genes, but they might produce different milk production or components, and epigenetics might be having an influence on that in some way, shape, or form. So it really brings it into an environmental influence, which we all do every day, which is eat, right? We all eat. So these mice here are, I don't know if you believe me or not, are actually all genetically identical. And they're actually raised within the same environment as well too. The only differing factor between them is that their mothers were fed either a high concentration of folic acid or a low concentration of folic acid. And so you can see this difference in phenotype from a very obese individual that has a different coat color, and as that concentration of methyl groups increases, you see that change in phenotype, and that's all just due to epigenetic differences on that gene. So to bring it into another aspect, it's really looking at these early life experiences. So early life experiences can really dictate that future phenotype for that individual. And this can be done through nutrition, exposure to toxins, or stress. So this really brings us into talking about the Dutch hunger winter. So think of World War II, it's Holland, and there is disruption in rail service. And this disruption leaves parts of the country in famine. And what they see is that babies that were in gestation or in pregnancy before this famine have differing have different traits than those that are within the pregnancy within that famine period. So one of the most starking differences is that they had lower birth weights. Well, that's probably due to the fact that mom had lower nutritional availability at that time. But when they look at the next generation, they see that same effect. So it's not just within that one generation. The next generation also has that lower birth weight. And this graph here is showing that the disease occurrence between these two individuals is actually quite different. So those individuals that were in gestation during this period of famine, so their mother had restricted nutritional access, you can see that they have higher disease occurrences, and that's including heart disease, obstructive airway pathways, and just overall general poor health, which they were rating upon hospital visits. And once this period of famine was gone, this effect was not in the population anymore. So it's just a little bit of food for thought. You can look it up if you need more information on it. So now to bring us to a rat study, and this is one of my favorite studies. It does come from a Canadian group, no bias at all, but this is done from a group out of McGill, and it's looking at grooming mothers. So what they did is they selected individuals that were either high grooming mothers or low grooming mothers. And what they were looking at overall was the stress response that those offspring had. And basically what they do is they put them on this little spool in a pool of water, and it's a very stressful situation for a rat. They don't like water. And then they were looking at the cortisol increase, so basically the stress response that those individuals have. So what they see is that those low grooming mothers have a higher stress response than those that are with mom and are being groomed constantly. So you're saying, well, they selected, high grooming, low grooming, there might be some of a genetic effect there. So then what they did is they actually switched them. So they took high grooming offspring and they gave them to the low grooming mothers. And what they saw is those high grooming offspring that had that decreased stress response, when they were given to the low grooming mothers, actually had an increase in the stress response, and that was maintained throughout that generation. They've been doing some work in looking at the actual mechanisms of epigenetic effects, and it's singing quite purely with that, but that group is working quite wholeheartedly on that, and especially in terms that has a great influence on the human health as well too. So how does this actually work? Well, I'm not going to go into great detail about this because there could be an entire course about it, but there's a number of different modifications that exist. So DNA methylation is one of them, histone modifications, microRNA. So it's really anything that's not a base pair difference. It's only those modifications to DNA. And what it does is it actually compacts that chromosome so that you don't have the ability to produce those proteins anymore, or that you have a reduction in reduced proteins. So you can kind of think of it as a landscape. So if that little purple cell at the very top is a cell that can turn into any other cell type, it ends up in these microenvironments that tell that cell what to be. So it might become a B cell, a macrophage, or a fibroblast. If you think about it this way, if you know if you've ever seen CSI, right, they can take any sample of anything and figure out DNA from it. And that's really because every cell in your body, as opposed to gametes, have the same DNA. But the epigenetic differences are what making a heart cell different from an eye cell that are different from a muscle cell. So now how can we apply this on farm? The way that I'm looking at this is that we have a number of different management strategies on farm, and environmental influences and pathogen exposure, as well as toxin exposure. And really, that's going to affect the overall cow. But it might be more than that. It might be seeing these effects further on in generations, not only in the F1 generation, but the generation after that as well too. And this is really to do with the fact that those oocytes that would be in the production of that next generation are being produced during gestation of that animal. And so there's been a lot of work that's been done in looking at those maternal influences on that. And the way that I look at it is like very common is called fetal programming, but epigenetics is that toolbox that allows that to happen. And now there's more evidence coming out that this might not only be in females. I don't have any cow studies yet, but eventually I hope to see this. But in some of the mice and human work, we're seeing that the males might have influence over, that there might be a male influence over those epigenetic changes. So really what's happening, I think, is that the environment that the baby is going to come into, or the calf is going to come into, is being transmitted to that generation to predispose them to that environment to make them probably have the most survivability. So really the message is that what you do today might affect those generations of tomorrow, and that I think as we start moving more into this field, it's relatively still in its infancy, but I think as we move more into this field, we're going to find that a lot of the decisions that we make are going to be influencing those generations further down the line. Also, just very briefly about clones. So, there are also some cloning going on down in Brazil. And what's most interesting is that these clones actually carry different epigenetic marks as compared to their counterparts. And this is very unique. So what happens is that when there's an embryo and it's being produced, all those DNA, all that epigenetic information is ripped off, and then it's put all back on. But with these clones, there's certain genes that actually maintain these epigenetic marks. And a lot of the time it has to do with growth factor. So that, that's going to tell that individual how much it's going to grow. And so what you see is these abnormally large calves, and they often don't survive for more than a week after birth. So, there's some implications there as well with epigenetics. So some of the future applications, this whole concept of nutrigenomics, we had an entire lactation symposium about it. I really think that epigenetics is going to start falling into that, because the way I see it is that there's two types of epigenetics, a short-term and a long-term. Long-term going through generations, short-term is that individual. We have estimated breeding values in genetics. But how about we start thinking about epigenomic estimated breeding values? Start bringing in some of those environmental components. It could be a measure for behavioral studies, understanding development a lot more. So epigenetics is essentially this interface between genetics and genetics and environment. It's allowing that environment to transmit that information to genetics to make a stronger animal in terms of phenotype.